Alright guys, welcome back to Underground Science. In this video we'll be talking about Ataxia Telangiectasia. Alright, so this is relating to non-homologous end joining defect, okay, which is a method for double-stranded DNA repair, right? Double-stranded DNA repair. Okay, so let's go and get started. So first thing we should know, right, is that Ataxia Telangiectasia mutated is what the ATM gene stands for, okay? So we have a gene called the ATM gene, also ATM stands for Ataxia Telangiectasia mutated. This gene, if it's mutated, what happens is you don't get a functional ATM protein. Usually what happens is this gene is functional, so you get a functional ATM protein, okay? A functional ATM protein is important because it phosphorylates p53, which is a tumor suppressor gene or a tumor suppressor protein. So if ATM phosphorylates p53, it activates it, okay? Another thing ATM does is it phosphorylates BRCA1, okay? Let's see what happens when p53 gets activated or phosphorylated by ATM protein, right? If p53 gets phosphorylated and activated, it can act as a tumor suppressor protein, right? How, right? So if p53 promotes apoptosis, or what it does is it decreases the progress of the cell cycle, right? So if it promotes cell cycle arrest, right, or apoptosis, that's basically that's why it's a tumor suppressor, because it's suppressing the chance for a cell to start becoming cancerous and replicating uncontrollably, right? That's the whole point of p53. And if ATM phosphorylates BRCA1, right, then the BRCA1 gene, when it codes for the BRCA1 protein, right, remember, BRCA is involved in homologous recombination double-stranded repair. So again, that's what phosphorylating p53 and BRCA1 does. It activates both of these proteins. Both of these proteins, right, when it's transcribed and translated, right, both of the p53 and the BRCA1, both of these are really important for either, if we're talking about BRCA1, for repairing DNA, and if we're talking about p53, for making sure cells don't just become cancerous, right? So p53 is important for inducing apoptosis or decreasing the progress of the cell cycle. If there's something wrong with the DNA or some mutation that's occurred, that's that would cause the cell harm if it continued in the cell cycle. Okay. So that's that, and then now what we should look at is more things about ataxia telangiectasia, right? So it's autosomal recessive, means you need the ATM gene. You need both mutated copies of the ATM gene, okay, in order to get this disease, because it's autosomal recessive. So first things first, let's focus on the immune system. Again, when we're talking about the ATM gene, right, if you phosphorylate p53, you activate it, and p53 is good at reducing cancerous cells or killing cells that are killing or making cells undergo apoptosis, I should say. If those cells if there's something wrong in the DNA, right, there's DNA damage or some mutation that's gonna cause harm to that cell in the future if it keeps on replicating, right? So let's talk about the T cells in the thymus. So we know that T cells mature in the thymus, right? And so in the thymus, these T cells, as they're developing, it's normal for them to undergo double-stranded DNA breaks. Okay, it's normal for that to happen, right? We'll talk about this in a different video, but it's through VDJ recombination, right? Where they undergo double-stranded breaks, especially for the T cell, so the T cell receptor can have diversity, right? So you can have different arrangements of the T cell receptor so it can bind to various different antigens, right, when you're exposed to these antigens. So what happens is when your ATM gene is mutated, you do not have a functional ATM protein, which means you cannot really repair DNA as well anymore. And especially if we're talking about our T lymphocytes, remember we said double-stranded breaks are normal. So you can have variety when it comes to your T cell receptor. But if you're making all these double-stranded breaks, so it's normal, but then you can't repair it because you have an ATM gene mutation, that means that means that you're going to start undergoing apoptosis, right? If you have many gene mutations or a lot of DNA damage because you can't repair your DNA properly, that may trigger the cell to undergo apoptosis. So if your T lymphocytes, as they're developing and trying to, undergo double-stranded breaks, VDJ recombination, but then they can't rebuild those double-stranded breaks, then that's going to lead to apoptosis of your T lymphocytes, which means you're not going to have many functional T cells. And if you don't have functional T cells, right, which is involved in cell-mediated immunity, remember T cells are very important for B cells to produce antibodies like IgG, IgA, IgE. So if you don't have functional T cells, humoral immunity is going to be affected too. So all in all, you may not have as good of a cell-mediated immunity and a humoral immunity. Okay, so both of these both your cell-mediated immune system and your humoral immune system is going to be affected. Okay, so your B cells and T cells will be affected if you don't have many functional T cells. All right, because again, your ATM gene's mutated, right? If your ATM gene's mutated, you cannot fix the double-stranded breaks that your T cells are constantly doing so they can have variety for their T cell receptor. If you cannot fix the DNA, if you cannot repair the DNA, that's going to trigger the cell to undergo apoptosis, which means you're going to start decreasing the number of functional T cells. Low number of T cells means, well, you have low T cells, and also T cells are important for stimulating B cells to produce antibodies. So you're going to have low antibodies also, right? So that's not good. So that's what we should know for that so far, all right? And then, so that was just for your immune system, okay? But ataxia telangiectasia affects your cerebellum also. That's why you have ataxia, right? Loss of balance, coordination, gait, and so on. So how does it affect your cerebellum? Well, if you have an ATM gene mutation, that's again going to decrease the DNA repair process, because ATM genes are responsible for what? What's one of the things that they phosphorylate? BRCA1, right? BRCA1's important for DNA repair. If you cannot repair DNA properly, apoptosis is likely. But this time we're talking about the apoptosis of nerve cells that are in your cerebellum. Well, what's your cerebellum responsible for? Fine motor control, coordination, balance, having a steady gait, right? If your nerve cells start undergoing apoptosis because you cannot repair DNA in those nerve cells because of ATM gene mutation, then your nerve cells in your cerebellum will die, and then you will no longer be able to have fine motor control. You will no longer be able to have coordination, balance, steady gait, and so on. So it's going to be, decreased fine motor control, decreased coordination, decreased balance. You may also see dysarthria, right? This is all for the cerebellum being affected, right? You're going to see an unsteady gait, abnormal eye movements, and so on, right? And so that's the effect that it has. That's the effect ATM gene mutation has in the cerebellum, leading to ataxia, all right? And then last but not least, we see telangiectasia, right? Hence the name Ataxia Telangiectasia. So telangiectasias are basically small dilated blood vessels, and we can see them on the eyes or skin, right? And then just talking about the risks of cancer. So ATM, we said, was important. A functional ATM protein was important for DNA repair. And as we know, if you cannot repair DNA properly, you have an increased risk of cancer. Well, in ATM telangiectasia, your ATM gene's mutated. So then you don't have a functional ATM, which means you don't have proper DNA repair, which then increases your risk of cancer. Well, what types of cancer? Leukemia and lymphoma, all right? And another thing that can help in the diagnosis, it's not the one and only, obviously, but it's one of the things in the diagnosis that you may find helpful, is that people with ataxia telangiectasia may have an increased alpha-fetoprotein level. Okay, usually this is produced by the fetal liver, right? But as an adult, though, you shouldn't have an elevated alpha-fetoprotein level. But you may see an elevated alpha-fetoprotein level in an adult who has ataxia telangiectasia. All right? So that's basically it for ataxia telangiectasia and its relationship to non-homologous end joining double-stranded DNA repair. All right? And so yeah, that's basically it. I hope you learned something, and I hope to see you guys in my future videos. All right? Thanks for watching.